this, I'm going to start the recording. All right, everyone. So hello and welcome to this another AILS talk. And before we start and I go ahead and introduce the guest speaker today, let me say a few words about AILS.institute and what we are, what we do. AILS is an acronym for AI for Life Sciences, and it's essentially a distributed community. I myself, my name is Artur Yakimovic. I'm based in London, UK, and several other community members are based in other places like Switzerland. I think we're probably going to have new community members from Germany soon. And essentially what we're trying to do, this is a non-profit organization aiming to bring together specialists from both sides of the spectrum, be it research in AI, machine learning, and deep learning, and research in life sciences. We all want to see a little bit more of machine learning used in life sciences and vice versa, have more life sciences applications in the AI field. And therefore, we're organizing this talks. There is if you go to the AILS.institute, you'll find yourself in a forum that you can use to exchange ideas, do journal clubs, etc. And since recent, we have started this talk series, hosting various speakers and trying to exchange ideas and learn more about how we can use AI and machine learning in life sciences. So, okay, gonna get more participants right now. All right, so as we go on, and I would like to kick off this talk because the aim is to keep those meetings relatively short. Let me just take a moment and briefly introduce our guest speaker today. So today we're hosting Norma Rzepka, and Norman is a co-founder of Scalable Minds. His company develops webKnossos, a collaborative web-based annotation tool for large-scale 3D image datasets and services for automated segmentation of datasets. Norman has a background in computer science and is located in Potsdam, Germany. And today he's going to be talking about annotation techniques for large-scale volume EM datasets. And with this, I'm going to just keep on admitting a few more people. And with this, I'm going to just do the minor tasks on the housekeeping. Yes, while I'm doing that, I would like to remind everyone to ask your questions in the chat to kind of foster the interactive session after the talk. And I'm about to make Norman host for this, so feel free to start your few words of introduction or presentation straight away. The floor is yours.
Thank you very much. So let me start the presentation. So thank you very much for the introduction. As I already said, I'm Norman. I'm the co-founder of Scalable Minds. This is a short agenda. First of all, I will talk a few words about what the company does and why I know a thing or two about annotations. And then I will dive into dense annotations, then sparse annotations, and then talk a bit more about collaboration and also quality control. So Scalable Minds, we built webKnossos, which is a web-based tool for visualizing and also annotating large three-dimensional data sets. And we also offer image services for automated image analysis, automated segmentations. We work together with a bunch of institutes. Two of them are the Helmstedt lab at the Max Planck Institute for Brain Research in Frankfurt, Germany. And the other one is the Schäfer lab at the Francis Crick Institute in London. So webKnossos is a tool for looking at large data sets. And then this is a data set that some of you might have seen. So it's the full female adult fly brain. And as you can see, it's a 50 terabyte data set, and you can interact with it in a very fast way. You can zoom into the data very fast, and all of that is happening from inside the browser. It also has annotation tools, so you can do volume annotations. And it also has skeleton annotation features, which I talk about a bit more in a few moments. And it also has a collaboration features for larger projects, so something like project management features and user permissions and everything that you need if you're trying to do a bigger annotation project. So on the services side, we offer to do automated analysis for EM data sets from the very start, once you get your data fresh from the microscope. So we can do stitching and 3D alignment, and pre-processing it is required there. And then once you have that, we can also do automated segmentation on top of that. That could be something like nuclei segmentation or synapses classification, detection, and also type classification, all of that. And if you combine everything, then you could get out of that a connectome reconstruction if you have the right data for that. So I'm adding a few more people, or Arthur, is you doing that?
Yeah, no, just feel free to add them.
Okay, there we go. So why do we need annotations in the first place? There are a couple of use cases that come to mind, and some of them will give you direct results. Like, for example, if you do an annotation, then you can generate a visualization from that. Like, you could get a mesh visualization that you can then use to put in a report or in a figure of your publication. You can also, of course, use it for quantification, because once you have some annotations, you can do some counting, or you can do some measuring, or whatever you want. So there's very, very direct results that you can get once you do some annotations. And then the second category is more indirect results. And that is when you have a machine learning system in the loop. So you can use the annotations to train a machine learning system. That could be a segmentation or a classification. And you can also use these annotations for evaluation purposes of such a machine learning system. And these are two fairly different use cases, which I'll talk a bit more in a second. So let's dive into dense volume annotations. I'm sure many of you have seen tools like this, where you can have simple brush tools, and you can just draw into that. And in a three-dimensional setting, you would go from one section to the next and use the brush to draw in every section. So this is not a very fast process, but it is a very accurate process, because you're giving your full attention to every single section in your data set. You can make that a bit faster if you have dedicated hardware, like if you have a Wacom tablet and a stylus, that can be a bit faster, or you could also use an iPad with an Apple Pencil, and it would also be a bit faster. What you can also do in a three-dimensional setting is you can save some time by just copying the annotation from one section to the next. And the idea here is that from one section to the next, your data won't be dramatically different. So you could get away with just copying it and just doing some minor touch-ups here and there, and you would still get a very high quality annotation out of that. Even better than that is, or faster than that is, if you're using interpolation in between those sections. So this is a screenshot from ITK Snap, which implements this. And as you can see here, the annotator only drew some annotations, let's say every 10 sections. And then the tool is able to generate an interpolation of all of these annotations, and then you will get a full annotation out of that. And then you could again do a second pass to run through your data and do some corrections. If you're working with a machine learning system, then in some cases you may already have an over-segmentation. So an over-segmentation means that you have more segments per object than you actually want. So it has errors in that. And with a tool like webKnossos, where we have this merger mode, you can actually be very fast of then correcting these errors, and you will get a high quality annotation out of that. And the cool thing about this is that the segmentation in the first place doesn't have to be very good. So you could, if you're dealing with multiple data sets, then you could use a model that is trained on a different data set and apply it on this one, and then just do the touch-ups. And of course, this will only solve an over-segmentation, so it will solve split errors. If you have any other errors, then you can again use the regular volume annotation tools like apporaches or something. Another interesting approach is using interactive labeling, where you actually have an integrated machine learning system inside the annotation tool. And these are two screenshots from Elastic, which implement something like that. So the task in this screenshot is to differentiate the membranes of these cells from everything else. And what the annotator did is just drew some squiggly lines on some background here and on membranes here. And then what you can get immediately is a live preview of the prediction that you would get out of that. And this is great, because then as a user, you can immediately see where the system will need some more annotations to actually perform better. So what I would do now is basically would add some blue lines over here, this area, and maybe some yellow lines to mark the membranes over here or here. And then do another round. So this is an iterative approach. So you can, after a few iterations, typically you can get fairly good results. If you want to get perfect results, then it will take quite a bunch of iterations to get it really perfect. So that is something to keep in mind. And another issue is that this works best if you can oversee the whole data set for yourself. If you can see all the spots where the prediction might not be optimal. So this works very well for 2D data sets or for smaller 3-dimensional data sets. If you're looking for big ones, then there's an interesting concept out there that is still in active research, which is called active learning. And the idea here is that the machine learning system will actually tell the user where it wants some more training data, and then the users can add more there. So you will start out with some basic annotations, train a model from that, and then the machine learning system will not only produce a segmentation or a prediction, but also will give you a confidence score. And in areas of low confidence will basically be fed back to the user. So there you can then do some more, add some more annotations, and then train again, and then again get some confidence scores. So this will be, again, a very iterative process. And the cool thing about this compared to a tool like Elastic is that your data sets can be very big, because the machine is telling you where to add some more training data, and you don't need to do that task as an annotator. But as far as I know, this approach is not yet implemented in any ready-to-use tool for large data sets or large 3D data sets. So now let's talk a bit about sparse annotation methods. because sparse annotation methods might not be or might give you good enough results and are much cheaper and faster to obtain. So for example, visualization tasks you can do, some visualization tasks you can do with sparse annotations. Think of like a density distribution of some nuclei in a data set. That could be something that you can get away with just sparsely annotations. Also, quantification is something where sparse annotations can be very powerful. Like for again, the nuclei example, if you're interested in counting them or like calculating some densities among different layers in your data set, it's something where you only need, let's say, one dot or point per nuclei. So counting is great. You can also do measurements with some simple lines or bounding boxes that you can draw. On the machine learning side, we typically don't use sparse annotations for training machine learning systems. So that's something where we almost always use dense annotations. But for evaluation purposes, sparse annotations can be very good and can sometimes even provide a better picture, because it's actually feasible to get a global view on your data than it would be if you do some dense annotations. So what do I mean by that? So for that, let's talk about skeletons, which is a sparse annotation method that is very popular for neurons. So it's line segment annotations. You have a bunch of nodes in here and all of them are connected with edges. And then you get this graph-like representation. And the cool thing is that you can get this for the whole data set or for the neuron spanning the whole data set fairly cheaply. And this will provide you a global view of where your data actually is. It actually has been quantified, so you can get up to 150 times faster than doing dense volume annotations of these neurons. So it's really worth it. And when we do a segmentation of neurons in a whole data set, then we typically use these because it will tell us where our neurons have some splits in there or where they merge with other neurons. And this will provide basically a global view. If we would only use a small but densely labeled bounding box, that would give us some information about how many pixels are off. But that's not really what we care about, because we really care about these global effects or these larger effects on the segmentation of these neurons. In webKnossos, there is this feature called the flight mode, which has a projection of the data onto a sphere. This makes it easier to move around in the data. And then combined with the fast data loading capabilities of webKnossos, this allows you to get very fast at generating these skeleton tracings. So if you're interested in neuron tracings, then that's certainly something I would recommend checking out. Other areas where you can use sparse annotations are for evaluating classifiers. Here on the left-hand side, we have an example of a nuclei classifier. So we basically get a score per voxel from our classifier, and then it was very easy for us to define a bounding box and go through that manually to mark all of these nuclei with just a simple dot each. And this will give us a good idea to figure out how many nuclei were picked up by the classifier, how many got left, so we can really get a good understanding of that. What it won't tell us is basically if we get every voxel of this nuclei right, or if we get or the contours are perfect. But for our use cases, that was not necessary. So that was a very fast way of getting some evaluation data for this project. And on the right-hand side, you can see a similar annotation where we use small line segments to mark synapses. So this is from a synapse classifier, and you can see in the middle, this reddish tone here is actually the synaptic cleft that has been picked up by our classifier, and we mark the cleft with one dot, and then to the left we have the presynaptic part, and to the right we have the postsynaptic part. And with this, these three items, we can get again some good evaluation on how well our synapse classifier works, how many of these synapses we found do they connect to actually the right neurons? And again, because it's only sparse annotation, we can get very fast results. So sparse annotations can be a very powerful tool and are quite cheap to obtain. If you want to be very fast and have to do dense annotations, then collaboration is a good way of doing that. So the main idea is to divide up your work. And there are two ways that we typically do. On the left-hand side, you can see you just take your data set and split it up by bounding boxes. And then you would take each bounding box and assign it to one annotator. And the great thing about that is that you don't need to know much about your data set up front. So you can just take your data set, chunk it up into distinct bounding boxes. Of course, you don't have to take your whole data set. You can also just sample from that if you're looking for training data. But again, you don't need to know much about your data set up front. If you do know something about your data set, then you can also split the work by objects. So one example for that would be if you want to do neuron tracing, manual neuron tracing, and you already have some seeds, for example, some nuclei, then you can just take all of these seeds, assign each seed to one annotator, and then have them do all of that. Typically, when we do training data for, let's say, a segmentation, then we go for the left approach, just by getting some bounding boxes annotated. And if we are looking for sparse annotations, then typically we look more to one on the right. If you are doing bigger projects for annotations, then some tools for actually managing these projects are quite helpful. And this is why we built a task management system into webKnossos. And the workflow works as follows. So it starts by creating some task descriptions. And we found that short videos up to 10 minutes or so work very well, because then the annotators can see exactly what the task is that they will be doing, and can also get to know the tool and see all of the details that are required to perform the task. The annotators can then log on to webKnossos, and there's a button for getting new tasks. And in the background, there is a task assignment system that has the whole pool of available tasks and will assign these tasks to those annotators dynamically based on groups that you can predefine, or based on experiences that the annotators may have. And then for an admin who has actually submitted the project, there's also a monitoring tools, so you can get an understanding of where your annotation project is, how fast it might be completed, and how much time it already has taken to do. And then, of course, there are also features to then collect all of the data into one workspace, and then downloading that data or working with that directly from webKnossos. When dealing with collaboration, though, there are a couple of things or caveats to keep in mind. And the first one is that multiple users will annotate the data in slightly different ways. So for example, if you look at these two examples here, this cell over here, or this object over here, cell looks a bit different than the one on the right hand side. So the difference is not dramatic, but it is still something to keep in mind, and something that we need to deal with. And the reasons for that are manifold, right? So it can be either from the data itself. Sometimes it may be hard to see where the border really is of the object that I want to annotate, or where the membrane is, or whether to draw onto the membrane or stop right before the membrane. So there's some ambiguity in the task description. Sometimes it's hard to see due to resolution of the data. And then, of course, there are also other reasons that lie within the annotator itself. Some give it their full attention, and some are already in a lunch break. So that's definitely something to keep in mind, and something that we then will need to deal with post hoc. But not only multiple annotators annotate data differently, even if you give the same task to the same person, you will also sometimes get different results. And you can see this this place. So this is but done by the same annotators. It's the same data, as you can see, and still we get some slightly different results. In this case, the data has been annotated in different viewports. If you remember webKnossos, in the screenshot there, webKnossos has these 3 different viewports. And for this project, we asked them to trace first in the YZ viewport and then in the XZ viewport. And as you can see, the results are a bit different. And again, now this is something that we need to consider when moving forward. Why do we actually give the same task to different people? Well, for redundancy purposes, because if we give the same task to multiple people, then it will help us with dealing with quality issues. For example, if people didn't have attention or did do some mistakes, then redundancy will help us. So yeah, the idea is basically you give the same task and give it to multiple people. So 2 would be a low number. Typically, we have 3, 5, or 7-fold redundancy, depending on the difficulty of the task and also what kind of quality requirements we have. But this also means now that with redundancy, we actually need to deal with that. And for volume annotations, what we typically do is then run it through an averaging. We can also do voxel voting and then do some connected components analysis on top of that, or you could do an intersection of all of that. And that really depends on the use case and also on the data that the annotators provided. So this is for us, this is typically a case-by-case decision that we need to do. And also for if you're dealing with skeletons, there's also a consensus protocol that has been published by Hamstetter et al. So that's also possible to get some consensus out of sparse annotations and to get all of the benefits from that as well. So with that, let's go to a short summary of what we've just been talking about. First of all, I've been talking about volume annotations. And if you want to get some volume annotations, then you can use some simple brush tools. There's some tools that make that go faster through interpolation or through copying of sections. If you are dealing with a machine learning project, then you might have some over segmentations that you can reuse to actually get some better or get some results faster. And then there are also tools out there that integrate a machine learning system into the labeling tool, and you can use those to get some fairly good results in a short amount of time. But not always do you need densely annotated volumes. Sometimes you can get away with just sparse annotations. And if that's the case, then I would highly recommend going for that. And for the particular case of neurons, and skeletons are a great way. And for others, you can get away with, you can use other sorts of sparse annotations. If you want to go really fast, then collaborative annotation is a great way of doing that. So divide up your work, think of how you want to set up your project and your task management, but be aware that once you get the results, there will be ambiguity in there, there will be variance in there, and to deal with that, redundancy helps with quality issues, and consensus is something that you also need to implement then if you use redundancy. So with that, a few more words about webKnossos. So you can sign up on webKnossos.org and just try it out. We also have a pilot program which will provide early access to new features, and it will also be looking for feedback. And you will also get a bit more storage to upload your data sets there. We also have annotation services. So if you're looking for an annotation project, then we can help you with that. And as I mentioned in the beginning, we also have automated image analysis services. So we can also do segmentation projects and help you with large image data sets. And if you have any more questions after this talk, then feel free to reach out to me. And with that, I would like to thank you for your attention and would open up the Q&A.
Fantastic. Thanks a lot, Norman. What a fantastic talk, and really great to see you finally tackling the consensus and the annotations across different people. That's definitely a useful tool and useful to learn about your experiences here. I think we've got a question from Martin on the chat. Martin, would you like to unmute yourself and just ask?
Yeah. Hello, can you hear me?
Yeah.
Sure. Yeah.
Okay. Yeah, thanks for the talk. Very interesting. can you, when you've got multiple annotations on one region, I guess that gives you some information. If everyone agrees exactly, then it was an easy bit. If people don't agree so much, it's a bit harder. Is that information you can extract and use?
Certainly, yeah. So we haven't quantified that in the project yet, but yeah. So typically, where we see more agreement is really where the image resolution or the image quality is much better. And if that's clearer, then that's will also be something where the machine learning system later on will perform much better. So that will give you an indication. But still, you want, but still, we typically want to sample regions from the data set that have multiple levels of resolution in them, so that the machine learning system can also deal with regions that are a bit more ambiguity from the data itself. So it makes the job of annotation a bit harder, but it also will give you some benefits down the road.
Awesome. I wonder, actually, since you mentioned ambiguity here, is there any, let's say, more formal way of tackling the ambiguity in the data as such? So I imagine that, you know, on minor things people can agree or disagree, but if there is no ambiguity, there is a way to just, you know, perhaps statistically address the you know, different opinions there. But what if data is very ambiguous? Is there any, let's say, and specifically for the situation of sparse annotations, is there any statistical way to address it?
So yeah, that's not something that we have looked into in depth. So I don't have a good answer to that.
I suppose perhaps when you will have large data sets of different collaborative projects, maybe there'll be a way to do some sort of a meta-analysis of how agreement happens and or how disagreement happens. That will be interesting.
Yeah, and I'll come back and give another talk.
You'll be most welcome to. All right, are there any other questions in the audience? We only have just a few minutes left. Don't be shy, feel free to unmute yourself and just shoot. Okay, Benjamin has a question, I think. Feel free to say, or would you like me to read it out? Okay, sure. So Benjamin is asking, he's thanking you for the talk and is, okay, nice. Would you consider your tool useful as a basis for deep learning approaches?
Yeah, so absolutely. So we use webKnossos for all of the deep learning projects that we do internally, obviously, and it helps you at multiple stages of that development process, right? So it can help you to get that training data. Again, we can split up the work and collect it back through the task system. So that's something that's very helpful. But then also when you have some first versions or something, then you can visualize that. So here we have the screenshot. We have a segmentation map that was created by a deep learning system. It's also flexible enough so that you can upload prediction maps. So you can look at the direct or the predictions that are the basis of this segmentation. And we also use it for error analysis at multiple stages, right? So for getting evaluation data, but then we can do some automated evaluation, and this will give us some errors, and then we typically load them into webKnossos to do some visual inspection to actually figure out what are the errors, do some error classification, and for that we find the tool very useful. So yeah, it helps in a bunch of stages, and of course it also will help you then once your data is all ready, then you can publish that data and share it with others. And some people even put links to their data set on webKnossos into their paper so that other people can browse it directly and see, explore the figures that you otherwise will only have in static form on your paper.
Oh, well, super. Okay, so I guess unless anyone else has any questions. Guess I would ask you then maybe.
I think there is a question. Yeah.
Okay, go ahead.
Sorry. So hi, I'm Jared. I'm from the EBI. So I'm living in the world of archive. So one of the things we have started to archive is volume data. So I'm still trying to wrap my head around things. So I'm trying to understand if we mean the same thing by annotation. So for me, annotation means taking a segment and attaching a label to it that has biological meaning. So ideally something from an ontology that says this is a ribosome or this is a nucleus, and because that helps us to link volume data, for example, to high resolution crystal structures or sequence databases, or whatever. Is that, do you do that as well, attaching labels to say what segmented features are? And do you use ontologies? If that's the case, do you use ontologies for that, or do you have your own terminology?
Yeah. So we do that occasionally. For example, when dealing with neurons again, then what we oftentimes do is running a type classifier on that, and that will give us some information about basically what part of a neuron we're currently looking at. For example, is an axon, is an a dendrite, or a nucleus or something, and then we can attach that information to a segment. So yeah, that's something that we do, and also then we'll help for other parts of even of the segmentation process itself.
All right, everyone. And with this, sorry, unfortunately, we're almost out of time. And I would just invite you to reconnect with Norman and follow up on this. And I would like to thank Norman, and I would like to thank you all for attending today. So yeah, thanks a lot. And if you would like to give a talk like that, please feel free to contact me. And looking forward to catching up with you in the AILS.institute forum as well. Okay, thanks everyone.
Thanks. Bye.